said, is your ability to compare two sequences to find what is similarities and to find the differences between these sequences will make you allow your cognitive skills to be much higher. Okay, let's get on. I'm not going to describe or give away the answers for what, but I will tell you what all you need to look at when you're trying to answer work for this towards this assignment. The first one is you can always go to for comparing quickly for sequences if there are any differences or not there or not. That is how you can go. Go to your sequence, something like this. The link is already provided to you. You go there and you put in your sequence here, protein sequence or nucleotide sequence. You can choose based on that, and then it will give you some. Here you should go to DNA, and then it will provide you what you can. Then the sequences. The website will show you the pairwise alignment. I think some of you have already done bioinformatics. That's an important thing, important course to have. And if you have done this, you will get a preliminary idea about what you want to do. Okay? Yes. Then what we will do is to find the length of the DNA. How do you find the length of the DNA? DNA is, I hope you know the structure of it. It's a coiled coil, something like this. And you have turn, and these are the hydrogen bonds between the bases that are present. And then this is the minor groove, and this side or this part and this part will be the major groove. So the length of the DNA, if you remember earlier from your basics, sorry, you have this from here to here. If you have this, and then you can actually, one second. Okay, sorry. Yes. So one turn from here to here, it is one turn. One turn, what is the size of it? You can actually keep messaging if you know the answers. 3.4 nanometers. It can also be said as 34 angstroms, right? You should, each of us should be good with the conversions from units, various units, especially with the length. And then how many bases are there? If it is there are 3 different types of DNA types, structures, A, B, Z, and you also have left hand, right hand, and so on. Those things you can look up. In bDNA, there are around 10.5 base pairs per turn of DNA. For calculation purposes, you might sometimes have something like 10 is approximation is fine, but it all depends again. Okay, so the question is about millimeters. Okay, you have to know the number of bases of your DNA, base pairs or bases, base pairs assume, and then you know from this you can calculate the length in 3.4 angstroms or so. Sorry, 34 nanometers, and then you can then calculate that into millimeters. Please answer only to the question. At the end, you should always put in mm and then write the units properly. The next one is about molecular weight. So the average molecular weight of a base pair is around 660 daltons. It all depends on what you're looking at. For AMP, it could be around 330 or so. And find out the average molecular weight of a base pair, and then try to do it. If one wants to be very accurate, the best way to do is what is the molecular weight of GC, you find out. what is the molecular weight of AT? Find out. Then when you calculate the number of bases, number of G's and C's, and number of A's and T's, the base composition, you can then calculate much more accurately. You can also compare the molecular weight of the DNA. You can Google up, many websites will be there. Find the molecular weight of the given DNA molecule. You give that, and then it can find the molecular weight. Just ensure you're asking for double stranded or single stranded. Just Google up. This is not a spoon feeding session. It is rather to guide you to the path. From there on, you have to take and explore many more things. In the pursuit of your answers, you will find more knowledge, and that's the objective of this whole exercise. And GC content is easy and straightforward. And the single stranded DNA that is given, all of DNA that is given to you is single stranded. Please note that. And then ensure that you're going to do, this is a single stranded DNA and the other one is single stranded DNA. You are supposed to convert it into double stranded and then work upon it wherever required. Okay, so you can find out the GC content. If it is single stranded DNA, you will not have the G equal to is not equal to C, not necessarily, and A is not equal to T. Or it's not that it shouldn't be, but there is no obligate rule that they have to be equal. But if it were double stranded DNA, then G must be equal to C and A must be equal to the number of T. Okay, and just for a quick thing is that G base pairs with 3 bonds, 3 hydrogen bonds with C cytosine, and adenine binds with 2 hydrogen bonds to thymine. I hope these are all fundamentals and you find that all easy. That's good. But when you're working, I would like that you are doing much more elaborately. Okay. So the next, as I was continuing in the previous, it's about you have for sequence A, how many hydrogen bonds are totally there between these 2 strands. And then this is sequence 1, and I gave you 2 sequences. So you are supposed to tell me what are the number of hydrogen bonds. So the length and all these things, everywhere you're mentioning, like in the previous one, you should always ensure that you're comparing the sequences. Okay. Sorry, excuse me. And then you have topological changes. In the last one, in topology, I'm not sure how much you have read about it. We have turns and linking number, right, and so on. We are not going into all the details, but what I want you to understand is twist. If this is one strand and this is the other strand, in a coil you are having like this, right? Each time one strand crosses over the other one, it is one twist, right? The twist is almost equal to the number of turns, right? And then that is what in a what is relaxed DNA, what is supercoiled DNA. All the DNA that I've given you is linear DNA. So it's unlikely that they are going to have either positive supercoiling or negative supercoiling. Assume that that is relaxed DNA. In relaxed DNA, twist is equal to number of turns, which is again almost equal to the number of amount linking number. Try to get these terms properly so that you will have a better understanding of things. The other one is nucleosomes. So I hope you know the sequences that I've given you are actually prokaryotic. But then, just for our understanding sakes, I hope you know what are the nucleosomes. There are 5 different nucleosomes: H1, which is a linker histone. So there are 5 histones, right? And then you have H2A and then H2B. These two form a dimer. And then we have H3 and H4. Okay, so these are core histones. So if you have per nucleosome, in one nucleosome you will have a total of 2 H2A H2B dimers, and then 1 H3 H4 tetramer. Okay, and about histones, they are rich in lysine and arginine, and they are around the that is what they are on the surface of the nucleosome. They are usually basic amino acids. Why should they be basic? That is because DNA is negatively charged. Why is DNA negatively charged? Because there is phosphate. Okay, the phosphate is highly electronegative, and if a protein has to bind to DNA, and then it must be having a lot of positive ions around. So nucleosomes are usually on the surface of it. They are rich in arginine and lysine amino acids. Okay, I hope I made this quickly, made you recollect some of the things. And in the last, you have to calculate the total number of nucleosomes that the DNA sequence that is given to you. Say, for example, I think it varies from organism to organism, but around approximately about 147 nucleotides. So assume that this is the DNA molecule. 1 second, give me that. I'll do that. So if you assume that this is the nucleus, this is DNA that is wound around nucleosome, it takes approximately 147 base pairs in humans. Okay, in yeast and other things, there are variations to it. Assuming that this is human DNA, how many nucleosomes would be required to bind the whole sequence of the sequence that is given to you? Okay, if you want to make all the nucleosomes bound like this, then how many? First of all, you have a DNA. Say, for example, it is around 730 base pairs. Okay, and if you have 730 base pairs, you can calculate by using 1 second. I can't find it, I think. So it will take time. I will use my mobile. 730 by 147. That would be 1 second. Sorry. I'm getting a call too. I'm unable to move. Yes. Sorry again. Yeah, this is what will happen with a lot of technology. I think my computer is slow. Anyway, I think I will just move on so that I don't waste time. So 730 by 147 should be the number of nucleosomes that are required. wind up the DNA that you have. So you need to calculate for both. And then, say for example, there are around, say, 5 nucleosomes that are required approximately. Then you would need 5 into um, you need to calculate the composition of each of these histones, and that can be done only that can be done if you are able to calculate the number of bases. Okay. And then I hope you understood in the previous one, what all we know is, what all we try to do is to be able to calculate or analyze the DNA as such, as a physical entity. How does it fold, structural, base composition, hydrogen bonds, and all those things. From now on, what we will do is to be able to understand the information of it. Okay. So the first one is, I hope you know that this is the, assume this is DNA, and an open reading frame is represented something like this. What does this arrow indicate? The arrow indicates that this is the start and this is the stop. This is the orient of the reading frame of a ORF. What is an ORF? Open reading frame. That means it starts with a start codon and ends with a stop codon. What is the start codon? I'm writing in terms of DNA, so I will leave it as ATG. Stop codon, there are 3 different stop codons. It could be TGA, TAA, and TAG. And this is how typically a promoter is represented as. This is the same kind of same way we will be using in most cases. So I would want you to pay attention to the way we are trying to represent stuff. Okay. And then there is, in prokaryotes, if you're going to see, there is -10 and -35. What is -10? If you can recollect, the sequence is TAT, AAT. That is one of the most optimal -10 sequence. But different genes have a little bit variations of this. And there are also different other promoters as well. -35 is TTG, ACA. Okay, that is the sequence that is optimal, but there are again variations. Sometimes you have up elements, sometimes you have extended -10 sequence, and so on. There is another sequence somewhere here, and that is called as SD sequence. That is for the ribosome binding site, RBS. Shine-Dalgarno sequence, that is where when upon transcription, something mRNA would be formed something like this. This is the SD sequence. That is where the initiation of translation would start. First is the ribosome binds to the SD sequence, and then it comes to the start codon, and from then on elongation will happen. In eukaryotes, the ribosome binding site is called as Kozak sequence. I hope you know that already. But these are some of the things that you would want to remember. So within the DNA sequences that I've given you 2 sequences. You are supposed to identify, first thing is to identify the number of ORFs that are present. Say for example, when you are looking for ORFs, I have already given a website. It's an ORF finder. You can go there and paste your sequence. It will tell you what are all the ORFs. But don't check something that is less than less than 10 amino acids. Ensure that it is, maybe you have to set a value to that. A ORF can be like this. All right. I could have used my sequence, but I don't want to, because I want you to explore your sequences. And that's the only way of learning it. So here is one ORF. You may also, because you have ATG here and TGA here. Sometimes what may happen is find another sequence that is starting another ORF start here and ending here. This could be another open reading frame. So these are overlapping open reading frames. And if I didn't mention some things here, so always every time you are representing, always ensure that you write what is 5 prime and which one is 3 prime. The polarity of the DNA strands should always be mentioned. Otherwise, we will lose something about it. Clarity of thinking. Minus 5 dash. Some people say that dash is wrong. I think dash is this. A prime is this one. So call it as 5 prime. 5 prime to 3 prime. So in science, it doesn't matter what we like and what we feel comfortable about. We have to follow these standards. That's important for science. 5 prime to 3 prime. And ATG you have here is one open reading frame. This is the second open reading frame. I'm talking about ATG present on the upper strand and TGA and so on. What about the bottom strand? This is assumed that this is the bottom strand. It will be it is anti-parallel and it is complementary to the top strand here. Like, so what if you have ATG on this one somewhere here, or sorry. Yeah, say for example, you have ATG here and TAA here. So this is a start codon. Is that right? No, this is not a start codon, because every time we read anything, it should always be from 5 prime to 3 prime. Every reading, replication happens 5 prime to 3 prime. Transcription happens 5 prime to 3 prime. Ribosome, the translation also happens 5 prime to 3 prime. That polarity has some meaning, and that's why we have to always be mindful of 5 prime to 3 prime. Right? So this is not a stop codon. Its actual sequence, because you have to read from 5 prime to 3 prime, it is not TAA, it is actually AAT. Here this is GTA. On the complementary strand, I mean, it all depends on where you put 5 prime to 3. You should have this, and this is one open reading frame. Here you have ATG. I'm reading from 5 prime to 3 prime. ATG, and then I'm ending with TAG, right? So keep that in mind all the time, so that you'll be able to look at the sequence much more critically, and then you can derive more information. So in this part, what I want you to find out is, within the sequences, 2 sequences that are given to you, find out the open reading frames. How many are there? And find out what is I mean, you should be able to draw a representative diagram like this, schematic, to say that from here to here I have one, there is another one on the sequence, and on the complementary strand there is another one. Okay, this is the structure that I've given here. This is of high gene density, meaning that within a small stretch of DNA you have open reading frames. So typically what you would have when you go into eukaryotes is, you will have one ORF there, another ORF there, and so on. You will have a lot of gaps within the regions. Whereas if you look at viral genomes, they have high gene density. That means within a small fragment of DNA, you have multiple open reading frames, which are actually functional genes as well. So gene density with viruses have the highest gene density, bacteria have next density, and from then on, with complexity of the organism, you find that they are very less gene density. Less than about 90% of our genome is just we don't know what it is. We'll just call it junk for the moment, right? Yes. At this stage, I would also want you to find the theoretical molecular weight of RNA, the complementary RNA. The chemically, if you're looking at the differences between RNA and DNA, you would find very few differences. DNA has A, T, G, and C, and it has deoxyribose. Okay, I'm trying to write what are the commons and differences between that. This is for DNA. And for RNA, it also has A. It doesn't have T. It has instead of thymine, it has uracil. It has G. It has C, and it has ribose. Right. So what is the difference between these two? So I don't have to bother about anything, except there are only chemical differences are actually only the ribose and deoxyribose, and the second one is thymine and uracil. And if you look at the thymine and uracil structures, you will find that uracil, if it has a methyl group on the 5' position, fifth position. Yes. Before I go, I would want to tell this. What is the difference between 5' position and the fifth position? If you're talking about 5' in nucleotides, that means this is the fifth position of the ribose or deoxyribose. I'll just write as sugar here. 5', the prime we are putting it to distinguish with the numbering from the nitrogen bases. Okay, so the prime, because I'm saying the difference between thymine and uracil is uracil. Okay, I should be able to make some space here. 1 second. So in uracil, uracil is easier to draw when something like this. You have N. You have a keto here, another one here. Solve for the double bonds. But this is something like uracil. Okay, 5-methyl uracil is thymine. That means this is just uracil, and this would be 1, 2, 3, 4, 5, and 6. If I have added a methyl group here, then it would be thymine, right? So the difference between these two, uracil and thymine, is the methyl group. Okay. Yes. So in the chemical differences, what you will find is that you have one methyl group is missing here, and or you can talk it as one. There is a need. You have an addition of a methyl group here, and here it is deoxy. Ribose is something like this, and I would want to draw this so that you will remember. The numbering is always important. So this is 1. I have to put prime here. 1, 2. I mean, you have to put prime if this sugar is already connected with the nitrogen base, because there you have another numbering. So I'll just put this as nitrogen base, and you have 1, 2, 3, 4, and this is the fifth one, CH2OH. Yeah. So you have something like this, OH, and the deoxyribose and ribose. The difference is that the second position, this is something like the ribose. Okay, and if you are having a if you are removing one of the oxygen here, then that is deoxyribose. The 2' oxygen is missing in deoxyribose. So the differences, chemical differences between DNA and RNA are very simple. That is the methyl group that is additional methyl group present in DNA, and the lack of oxygen in the 2' position of the ribose. And that's how you should be able to see these two. Okay, I think the last one is about you should be able to figure out a theoretical molecular weight. How can you calculate molecular weight of a protein? The simplest one is, I think some of you I might have discussed, you always have to remember this number. Sorry. 110 daltons. That is the average molecular weight of an amino acid. It's not any particular amino acid. It's the average molecular weight of any amino acid that we think. That's how it's an average. So it's easy to now calculate the theoretical molecular weight. It's not the absolute molecular weight. Absolute molecular weight will differ based on the composition. Theoretical molecular weight will be, if you know the number of amino acids, and then into 110 daltons, sorry, that will give you the theoretical molecular weight of the the protein in question. So each of these ORFs will have, you should be able to give how many ORFs are present in sequence A and how many ORFs are present in sequence 2. In sequence A, what are the molecular weights, theoretical molecular weights of the amino acids? The protein encoded proteins in both the cases, and that will allow you have a strong foundation in molecular biology. Then we'll look at, we have already seen, we came until molecular weight or open reading frames and molecular weight and so on. What we will now have is to little bit about translation, right? If you have number of ATPs, you should be able to calculate the number of ATPs required for the production of the protein. So in each of the cases, you have the pre-initiation complex formation. So you have, these are all in regard to prokaryotes. Keep that in mind. I know I'm asking about at some points I'm asking about eukaryotes, but majority of it is prokaryotes. So that's good enough for our understanding. So the ribosome has to bind for initiation. So you know the number of codons that are present, right? Assume these are the codons that I'm drawing here, and then you have a stop codon. For initiation, the ribosome should bind, and it requires a certain number of ATP. You look up the textbook and find it out. Okay, I don't want to say that. And the same way, for termination to occur, there are certain number of ATP. In translation, count all ATP plus GTP required. Okay, because they are all still phosphate bonds and they are high energy rich. And we want to calculate the number of ATPs consumed to produce a one protein, right? To translate one protein. The ribosome should bind here, initiation, and then for each codon, you have a certain number of ATPs required, or energy required, ATP or GTP. That is for tRNA charging. tRNA charging, you have EFTU, you have elongation factor small e, elongation factor TS, capital, I think. Sorry. Small e is for eukaryote. Okay. I think here it is EFTU and UFTS. They are important for the charging of amino acids, charging of tRNA with amino acids, and then bringing the tRNA to the ribosome, and then adding the ribosome into adding the amino acid to the growing peptide chain. So what we will know is that There are many more things involved. Peptidyl transfer center. You also have certain proofreading mechanisms. Translation is one fantastic course. I'll try to share my older videos on translation. I had that done, so I hope it will be useful to you. Just check it out. So you'll be able to calculate the number of ATPs required for the production of proteins. Okay. And then what I want you to also do is now the sequence of the proteins determined by the genetic code. So in that sense, what I want you to understand is genetic code is degenerate. In the sense, the genetic code is, you have to have a look at the genetic code. Some amino acids are coded by more than one codon, and some amino acids are coded by only one codon. Okay, so it's always a good thing to see. Sometimes, why I'm asking this particular question is, I have given you two different DNA molecules, and I might have changed in one of these, there is a mutation. But even though there is a mutation at the level of DNA, the encoded product may not have changed at all. And that is the reason why I want you to have a look at it in that sense. Okay. Then I want the empirical molecular weight. So once you have determined the amino acid sequence of the protein, go to a website, Google search how to calculate the empirical molecular weight of a protein, give the amino acid sequence, it will tell you the molecular weight. And then I want you to compare the theoretical value you calculated in the previous one. And here, I think you calculated the theoretical weight in the previous, and now you are actually using the actual molecular weight. So I want you to find out, compare these two and tell me what is the difference. And that's the way of, that is the proper way of analysis and looking at things much more critically. We are not doing anything superficial here. We are all in, and we will do it as much as possible. The difference between people, I mean, something is shown to you, and our ability to see beyond what ordinary people see is what makes us a scientist. We should be critical in the way and observe very keenly and try to be able to see what are the differences and compare, and this depth of observation should be very high. That is the goal of this whole exercise. Okay. Yes. Now comes the important part. That is, we have two different sequences, and there are some mutations. I'll quickly try to give you some overview on the different types of mutations, right? So mutations can be studied in several levels. I will try to finish this quickly. There are point mutations, there are large-scale mutations, there are chromosome aberrations, but they are not mutation, but there are aberrations, yes. In point mutations, you can have insertions in all these, in large scale or protein. You have insertion, you have deletions, you have inversion. What else? Okay, we'll limit that at this point. Insertions, deletions, inversions. You can also have duplications. That is also kind of insertion, but the same sequence is there. So duplication. And this is with the level of DNA. We are not talking anything about what happened to the information of it. We are talking it in terms of physical molecule, what all the things that happened. Insertion, deletion, inversion, or duplication, and so on. There are also other things that can happen. I mean, because of these changes, there is also a possibility that the information is changing. That is, you're trying to see it at the level of ORF, and because of which, in that, you can have missense mutation. and nonsense mutation. And there is one more important, that is frame shift mutation. Right? Missense is insertion, sorry, so I forgot to mention here, substitution. I forgot, that's important. In point mutation, there could be substitution, inversion, deletion, insertion, or duplication, many things like that. So the missense is like, I hope you know one more. In substitution, there are two categories. Can anybody say A is changed to T? What would it be? A is changed to G. What would it be? If A is substituted with T, what would you call it as? There are two terms, transition and transversion. I hope you recollect that. If adenine, sorry, if a purine is replaced with purine, that is transition. If pyrimidine is replaced with a pyrimidine, that is transition. If purine is replaced with pyrimidine or the vice versa, that is transversion. So in this case, this is the purine and here is the pyrimidine, so it is transversion. Purine, adenine, and here guanine, purine, that is transition. Right? Purine replaced with purine. Okay, coming back to this, missense is if you have a codon and because of the genetic code is degenerate, even though there is a substitution, there is no change in the amino acid that is coded, then that is called, those are called as missense or silent, sorry. Missense is because of the change, there is a amino acid difference. Say for example, there was glutamic acid and now it is glutamine. It has changed into the, when it is encoded as a part of protein, then it is missense. Here substitution is something like glutamic acid, because of the, even though there is a mutation at the level of DNA here, it's still, if the codon is still encodes glutamic acid, then that is silent mutation. Missense is glutamic acid being coded by, because of the mutation, it now codes for glutamine, then that is missense. Then there are nonsense. Any sense code that is now going to become a stop codon. Any sense codon that results because of a mutation becomes stop codon, then that is nonsense mutation. There are frame shift mutations. I think this will take longer time, but let's see. So here I quickly drew 3 sense codons, and this is a stop codon. So assume that this is a small open reading frame. If I delete this, now the coding region, the codon reading would be like this: CC, ATC, ACG, ACT, GA. This was an open reading frame. That means it starts with the start codon and ends with the stop codon. But because this G is deleted, now there is no start codon or no stop codon. The whole open reading frame has vanished. Right? So you have to, that is a frame shift mutation. A mutation because of addition or deletion of bases which are not multiples of 3. Why am I saying which are not multiples of 3? ATG, say for example, in this case, assume that G is fine, and I'm going to, just for ease, I'm going to delete this CCA. Then the sequence would be ATG, GAC, TGA. There is no shift in the reading frame. Okay. I hope you got that. Try to have a look at the frame shift mutation as well. In your sequences, you have to look through them and identify what is the difference, what has caused the mutation, describe the mutation in the at the level of physical like this, and then try to see how it has changed the open reading frame, describe the the mutation in the nature of the how genetic information has changed. And that is when you will also know the compare the open reading frames and see how the proteins have changed because of that. This is an important question. What may have caused the mutation? Say, what kind of things facilitate the mismatches? What kind of substitutions, deletions, what kind of physical, chemical, or biological agents cause or amplify or make help these kinds of mutations to happen? That is what you will have to, that's theory, have to have a look at it. If you are having a mutation that is if you are having a mutation, one second. Okay, I'm sorry. Yeah, I'm sorry. So look up in the biology, molecular biology textbook, and based on find out what is the what caused that kind of mutation. If it is frame shift, which kinds of physical or chemical agents cause, and so on. Okay, and then you will have yeah, and consequence of mutation that I already described. And I would want you to also tell me what kind of repair mechanism would have been employed if it is in the cell, if it is a mismatch that is being formed, what kind of nucleic acid, sorry, repair mechanism might be involved, something like mismatch repair, right? It is just to ensure that you understand what is happening. I want you to learn two concepts. One of it is called in terms of repair. You should know what is fail-safe glycosylase. I think you already know, but it is still worth trying to know. It's because I like it so much, because that's kind of how evolution is kind of smart and how smart things evolve. That's how probably I should put it at. And I hope you read about transposons. Transposons are jumping genes. And if there is a sequence like this, okay, there is a sequence. And okay, I will write the complementary sequence as well. And now assume that there is a transposon. I mean, I think you know that transposons are jumping genes, and they want to come, they are going to integrate here. Typically what they do is they cut the DNA something like this. So now you have two different sequences, that is GACT. And here you have CTGA. And this part I have to write, that is TCTG. Then I'm going to write this one on the top strand, AGACTAGA. And then I'm going to write this ATCT. Now the transposon has come and it got integrated here, right? And that's the transposon. Assume that this is a cut paste method. And once it is integrated, there are gaps here and here. So those will be repaired, and you will have addition of the sequence by the repair mechanisms A, C. and here you will have T, C, T, G. I hope you can see something new compared to this sequence. You see that this sequence was actually present only once here, that is this part. But now you have two pieces of the same here. That's called as TSD. That is target site duplication. Why I'm describing this is when you are comparing your sequences, you need to look for target site duplications. Say, for example, this transposon, because I said it is a cut paste method, gets cut here and jumps away, jumps out of it. Okay. Now what will you be left? You will have this region. Okay, we'll write it GACT and CTGA. Then you have AGAC and TCTG, and you have AG. I'm writing this part of it. AG, AC, TC, TCTG, and then TAGA, ATCT. Now, I hope you realize that now you have this chunk of DNA or this region represented twice. If you find a region, if you find sequence like this, what does that say? That means it was a footprint of a transposon, right? So look for your sequences and ensure that you identify these sequences properly. Not that everybody will have whatever I spoke. Different people will have different things. It all depends on how well you are looking at. So try to pay attention and find out these aspects. Last one, okay? I'll stop with this. So I think we are running. In PCR, upon denaturation, you will have DNA becomes like this. Now there are primers should be designed, and primers, if they are designed like this, if I put primers like this, is it right or wrong? And this will have 3' and 5'. This would be complementary, so 3' and 5'. They are wrong. Why is it wrong? As I told you in the early in the class, is all replication or all polymerization, nucleic acid polymerization, happens from 5' to 3'. Okay, so if I had designed primers like this is 3'. Polymerization, if it has to occur, it would have occurred outwards like this. That's not going to serve the purpose of PCR. If you didn't study PCR or you don't remember, it's a good idea to have fundamentals of it done. I am sure each of you are taught PCR, so no issues about it. Instead, what would have been correct is if you have designed primers with this way, so that you would have 5', and this would be 3'. Here is 3', and here is 5'. And now if you add DNA polymerase, this would have polymerized it in this direction, and this would polymerize in this direction. And now you have 2 DNA molecules, new ones. Okay, do it for 1 or 2 or open reading frames, the largest open reading frames. If you don't have it, do it for the whole sequence. And calculate the melting temperature. You can find the formulas of it. Okay, I will stop with this. You can also do these things and then try to find out the probable function of the gene. Each sequence I picked from a functional one. So you better try to find out what is it coding for. Try to read about it. The objective of this is simple. Imagine if all of you have done this well, made a video and posted it that can be visualized by everybody else. Within a week of watching all these, say if not a week or 10-15 days, we would have a large database of information, different varieties of information, and different ways of presenting different skills that you may have. And that's going to be very useful to everybody. And that's why this is, I'm very particular about this. All have to do, this is a compulsory assignment. At least in the 25 marks, I will give a big chunk of marks for this one. So do it well. This is a win-win situation in all the ways, because you will learn, benefit from it. You will be able to follow the rest of the course well, and at the same time you are also improving your presentation skills and a lot of technical things that you are learning. And that's why take it seriously and do it well. Evaluation will be well. There are also 3 more questions in this, I think. Homologous recombination and site-specific recombination. There is a Mendel's question, and there are also some topics where each are supposed to present things. So I'll give a good number of marks for this, provided you all are doing it well. Okay. I will stop now, and can you please have any quick questions, 3 or 4, if you have any doubts regarding the assignment? Hello, any questions? Is the time sufficient for the assignment? I think I set the time. That's one question I would want to ask. Yeah, tell me, Pushpali.
So regarding the deadline, you have mentioned it as Friday. Can you please extend it a bit, like maybe 10 days?
Total 10 days or 10 days more?
No, sir. 10 days from today.
Okay, I will do that. But see, one thing you have to remember this is the more amount of time, the less the productivity. Okay, but I will do it for you because you asked, so I will do that. Ensure that the quality is there. Typically, we wait until the last moment and then we do it. If we give you 10 days, 8 days of it is wasted actually. If I give you 4 days, you would actually work more. But anyway, I will do that. Yes, you have another question?
Yes, sir. Regarding the video that we have to make, it should be
yes.
It should cover all the questions, right, sir? Like all the questions in the same video.
No, just do for the first question and the last question.
Okay.
For Mendel's laws and homologous recombination or site-specific recombination, I don't need that.
Okay, sir.
Did you all understand that? Yeah. So each of you will make 2 videos. Each of you will make 2 videos. One is for the analysis part, the question number 1, and the second video is for the last one, where you have to talk about a concept. Okay? Yes. Gopika? Anybody else has any questions?
So this is Shweta here.
Yes.
So for the first one,
Yes,
the sequence analysis, do we have to put our answers in a form of a PPT and just take it as a, like you asked us to record the video? Do we have to, like, for example, you asked
That's
us to go and search in the internet for the databases. So should we also have to record that, or just the final answer?
No. Just the information.
Okay, sir.
Yes. You will calculate everything. You will have a handwritten note or whatever you do. You make that information into a PPT,
okay,
and then you record it using OBS.
Okay, sir.
So that I will know that you are presenting, and it is a good practice for you. Okay. We are all having, as teachers, I don't know about others. I myself, I am so, it is not easy doing this. And if you, at your age, get used to it, I think you will be very competent teachers with communication skills and so many technical skills as well. We are struggling to learn at this age now. Instead, if you get used to this, you'll become technically competent and self-confident. That's the idea.
Okay, sir. And also
Yes.
regarding the
Go ahead.
time extension, is that 10 days for everybody or only for those who ask?
Yeah, whoever gives me a chocolate. No, for
Okay,
everybody.
sir. Thank you, sir.